This week's episode is featuring Purple Cow's Bioactive Supercharger. Bioactive Supercharger is available in a liquid and a soluble powder product that provides your plant with living organisms and trace minerals to produce nutrient-dense vegetables and fruits. Visit stbiologicals.com to learn more about this and other regenerative ag products. Hello, and welcome to the ST Biologicals Soil Speaks podcast. This week, we'll discuss why plant tissue analysis matters and share how to read a plant tissue test. Let's get started. Plant tissue analysis is a powerful tool for assessing the nutritional status of crops. By analyzing plant tissues, growers and agronomists can identify nutrient deficiencies, excesses, or imbalances. But you have to know how to read a plant tissue test. The correct sampling techniques, optimal sampling timing, and the relationship between plant tissue and soil tests for testing all need to be understood to be beneficial for farm ecosystem health. Plant tissue analysis complements soil testing by providing real-time information on nutrient availability within the plant. Unlike soil tests, which assess nutrition levels in the soil, plant tissue analysis directly evaluates the plant's nutrient uptake and utilization. Here's why it matters. Early detection. Plant tissue analysis detects nutrition deficiencies before visible symptoms appear. Precision. It guides targeted fertilization, preventing overapplication and reducing costs. Crop-specific recommendations. Different crops have varying nutrient requirements, and tissue analysis tailors recommendations accordingly. Now, let's take a closer look at how to take plant tissue samples. We recommend regularly sampling throughout the growing season, about every 2 to 4 weeks. This will give you data for every critical growth stage from pre-bloom and fruit set to flowering or to fruiting. Randomly select plants from different areas of the field to ensure you collect representative samples. Avoid sampling near field edges or areas with different soil conditions. Be sure to combine multiple plant parts, such as leaves and stems, to create a composite sample. Once you have identified your sample plants, it is time to select the right plant part. Young, fully expanded leaves are ideal. Avoid old leaves that may not reflect the current nutrient status of the plant. About 20 to 30 leaves will adequately represent the entire plant. To take your sample, avoid contamination by using clean tools and containers. Avoid touching samples with your bare hands. Label each sample with relevant information, including field, date, and crop. Keep detailed records. You've identified, you've collected. Now it's time to interpret your plant tissue test results. First, take a look at the nutrient ratios. Compare nutrient levels, such as NPK and micronutrients, to optimal ratios. Imbalances, such as high N and low K, may indicate excessive vegetative growth, but poor fruiting. In addition to NPK, take a look at calcium, magnesium, and sulfur levels. Assess iron, manganese, zinc, copper, boron, and molybdenum concentrations too. Now compare your results to establish critical levels. Are they deficient, adequate, or even excessive? Remember, plant tissue and soil tests are complementary. Soil tests provide the foundation, while plant tissue analysis offers real-time insights. These tests help you correlate soil nutrient levels with plant nutrient uptake and fine-tune your fertilization strategy for better efficiency and higher profits. At ST Biologicals, our crop consultants have spent years guiding farmers and ranchers in reading plant tissue test results. for expert guidance from our knowledgeable team. Visit stbiologicals.com. When soil speaks, we listen. Thanks for listening, and we'll catch you on the next one.